welcome to 19 Washington Square North, which is, I've heard, the gateway to Abu Dhabi. So it's actually the building of NYU Abu Dhabi, and it's basically where a lot of things that allow us exchange between Abu Dhabi and New York. And actually, NYU Abu Dhabi today is providing us help for organizing this series of luminaries of science, and we have a few very bright people who came to give talks here, in general, when they give a talk in the afternoon, which is a public lecture, trying to reach a very wide audience. So today's lecture is by Denis Duboul, and Denis Duboul is somebody who is very important to me. He's actually a very good friend. He's also one of my scientific heroes. And Denis, as you will see, is not only a great scientist, he's also somebody who thinks very well and thinks straight and thinks science and thinks philosophy of science and thinks evolution. And I don't know exactly how he's going to present his public lecture, but I'm sure you're going to enjoy it. One of the claim to fame of Denis comes from his understanding of a set of genes called the Hox genes. So let me give you some background on that, and maybe you understand why this is quite important. So actually, when I started in science, I started a bit earlier, but then I really started in science, it was in 1984. I'm a bit older than that, but in 1984 something quite important happened, because in 1984 we discovered, people discovered what we call the homeobox, which is basically a motif. This is my seat. So in 1984, scientists discovered a motif, which is basically a piece of DNA of protein, which is going to was conserved between fruit flies, where the gene was first discovered, and human. And basically, what this piece of DNA was is actually a gateway, not to Abu Dhabi, it's a gateway to understanding development in vertebrates. Because until 1984, we thought people would study fruit flies or nematodes for the sake of studying insects, and this would not be very helpful to understanding what happened in higher animals, in mice or in humans. And then, when we discovered the homeobox, one discovered, in fact, what happened, the two species, the human and the flies, were speaking the same language, language of DNA. In the sense that the same basic principle that existed in flies, that had been discovered in flies, would also apply to humans. And in particular, this homeobox led to the discovery of a class of genes called the Hox genes. And the Hox genes are a series of genes that basically express along your anterior-posterior axis, and they tell you where each one of your body parts is. And one of the beauty of these genes is that they're all clustered on a chromosome. That means they're all next to each other, and they all control our development by the mere fact that they're all clustered together on the chromosome. And so Denis has been studying that, and over the last 20 years, he has basically provided 30
15.
He has provided deep insight in terms of how basically the chromosome act together to be able to control this expression. But again, Denis is much more than that. He's always thinking about how his work has implication in terms of evolution, and he's going to tell us today about understanding the molecular homunculus. Denis?
Thank you, Claude, for this very kind introduction. I'm touched, actually. I'm very happy to be here. I'm a little bit lagged, so if, let's see. We are February 27th, 2013, and actually I wanted to start my talk by showing you this picture. That was a paper published in the New York Times. See here, it was February 23rd, 1993, so it's almost day to day, 20 years ago, in fact. And this paper was entitled Making an Embryo Without Thank you. It was a paper written by Nathalie Mangier. You heard of her, at least for the oldest in the audience. Nathalie was reporting on discovery by many laboratories, which Claude rapidly alluded to, of a family of genes which are very schematically presented here. These several genes clustered on the chromosomes, and the fact that these genes would, in a way, represent the actual structure of the embryo, pre-represent the actual structure of the embryo. Now, there's something was quite interesting she wrote in this paper. She said, making reference to Star Trek, that was the right time, on the aliens of Star Trek, she said, well, they have bodies separated in two basic regions, head and trunk. They have eyes, they have ears, mouth, etc. They have arms and legs and tightly symmetrical bits. And then she went on saying, well, but lest the creators of the series be accused of intellectual timidity or laziness, it turns out that nature too believes in the all-purpose, ever-reusable blueprint for building mobile bodies. And actually, by reading this again, I was wondering last week before I came here, if there is a relation of causality between these two statements, in the sense that, is that because we cannot really imagine how to build a body in any other way that it is currently being done, that we are designing aliens the way we do. That's all the problem of free will, actually, and I'll come back to this at the end of my talk. Because I really like to talk about constraint, the notion of constraint, and this is what I'd like to address with the homunculus. Now, the homunculus was part of this paper in the New York Times, in fact. That was on page 2, and I actually don't know where she found this picture of the homunculus here, and she wrote, well, since this sequence, saying the sequence of genes, follows the body plan, it echoes the medieval notion of the homunculus, or miniature person encased in the sperm. Now I tell you, that was 20 years ago. People in the 18th century were more caring about the gender issue than she did, because it's not only in the sperm, it was half of it in the sperm, the animalculus, and half of it in the oocyte, the ovis. Okay. For example, the great French naturalist Buffon, he was convinced that cows were having the homunculus in their oocyte, not in the sperm. So there were the two separation into two parts of this preformation. Now let me tell you a few words on preformation versus epigenesis, because this is what it is about. That of course started with Aristotelian philosophers, and I'll come back to this later with the idea that development, evolution, I remind you that until the late 18th century, development and evolution was one and the same word to qualify a single thing, okay? That it all started with something that pre-exists, and what we do is only to sort of open, to reveal something that exists, okay? And that of course culminated during the 17th and 18th century, and I'm sure you've all seen this beautiful drawing from Nicolaas Hartsocker, this Dutch scientist, in this famous essay, The Dioptrique. Please note that at that time, the real science was written in French, not in English. It changed a lot. Preformation, see here, preformation had experimentalists, and in fact, probably the most famous one was a scientist from Geneva, who was a French emigrate, of course, as many scientists in Switzerland who came from France at the Révocation de l'Édit de Nantes, who were Protestant and they had to leave France. And his name was Charles Bonnet. Charles Bonnet was the inventor of the stairs, l'échelle des animaux, the échelle of animals, with the classification. Of course, we are to the very top here. But Charles Bonnet was a strong believer in preformation, and he decided to demonstrate that it indeed existed. And what he did was to carefully select green flies, virgin green flies, and he put them under a cheese cover, only females. And to his satisfaction, a few weeks later, he realized that there were plenty of green flies, and then he concluded that females do not need males, and therefore that preformation existed. In fact, he had been particularly unlucky to select one of the few species that reproduce via parthenogenesis. It's a true story. But all of this participated to the entertainment of this idea that the embryo was preformed. There were, however, people at that time and even earlier who had very strong opposition to preformation. And to me, the most brilliant was Pierre the Maupertuis. He was a great mind, which is not often referred to, but already in 1745, he strongly opposed to preformation, based on a mere watching of interethnic progenies. This guy, by the mid-18th century, realized that when people from different skin colors were having a progeny, usually, generally speaking, the progeny had an intermediate color. And he could not reconcile this with the idea of preformation. This is a fantastic observation at that time, okay, Monsieur de Maupertuis. Now, of course, along with time, along with the absence of any visible homunculus, you know, increasing optical tools, new microscopes, it turned out that it wasn't possible to see anything looking like a homunculus, and also because of the recombination of characters. Finally, epigenesis vanished by the end of, well, certainly by the end of the 19th century, but already in the middle of the 18th century. That, of course, brought a very complicated problem, which until then was not raised. It is the question of the origin and of the making of the embryo. From the time you decide that the embryo is not preformed, then you have to explain where it comes from. It's a different magnitude in problems, in fact. Now, actually, before that time, the embryo received very little interest. And even if you read the origin from Charles Darwin, there is a chapter on the embryo when he treats of variation. Darwin had a problem with variation, and there's very little pages, very few pages on variation. And when he talked about variation, he talked about the embryos, and he says, well, I'm sorry, it's in French. He says, actually, the embryology acquires a large interest if we consider the embryo as a sort of a portrait of the common ancestor. So he even considers the embryo as a sort of a proof of principle of the common origin. This is what he's using embryos for, actually. That's it. Not as a source of variation. Okay? It's very strongly influenced by von Baer and later on by Haeckel, with this famous drawing. Okay? Look, for example, here with invertebrates. You see these almost adult forms. If you go back into developmental time, you hit this stage here where all these embryos look quite alike. This is a drawing from Haeckel. You may know that there's been a lot of discussions on these drawings. He's been accused of adapting the drawing to his ideas, which is certainly true. If you look at this human embryo here, if you ever looked at the human embryo, that looks like a sheep, actually. Not at all like a sheep. However, the basic principle is totally correct. If you go backwards into development, you see that shapes tend to homogenize to reach a sort of a point where these embryos express the major features of their taxon, in this particular case of vertebrates. Now, interestingly enough, Haeckel and von Baer, they did not have very powerful optical tools, because if they would have been able to go a little bit before this stage here and look at early cleavage embryo, before gastrulation, before that particular stage, they would have realized that then the difference occurs again. Okay? So in fact, if you look within vertebrates, you see that early on, it's very different. The way the embryo go through cleavages, and those of you working with zebrafish or with mice or with snake embryos, with epibole mechanism, or with you find all kind of things. There's not a single embryo that produces the early material the same way. Okay? But at some point, these embryos have to go through a sort of a bottleneck here, which has been referred to as the phenotypic stage by Jim Smith and Peter Holland about 20 years ago. They have to go through this point here in a way to express their belonging to their taxon. Okay? And once this is done, then they can start diverging and acquiring these very highly adaptive traits.
The moment of gastrulation happens, yeah? This is right after, during gastration. It's not a point, it's a transition, okay? What is similar is not a snapshot, it's a process, okay? Now, of course, this point here can, in a way, be defined as the point of maximal constraint during development, okay? You can do a little bit what you want here. You can sort of diverge here, depending on the adaptive environment. However, here you are constrained to go through this point for some reasons, and this is what I'd like to discuss. It's the reason why this constraint exists. Now, the person who I think was the first to really clearly state the importance of the embryo, thank you, Claude, is Thomas Huxley. Thomas Huxley was really a genius. He is often presented as the person who was the best advocate of Darwin's theory. I would personally say that he was the person who explained Darwin, what his theory was about. Almost. He was really a brilliant mind. I'm kidding a little bit. But Thomas Huxley said, well, the theory of body construction, epigenesis, must complement the theory of change, natural selection. Okay? This is how to get large changes in anatomy. So he really is the person who said, well, wait a minute, embryology is important in the framework, in our understanding of evolution. You have to consider the embryo because this is where things happen eventually. And of course, about a century later, the genetics came and finally made the link between development and evolution, and this is what Claude was saying in his introduction. From the time where genes were isolated which controlled development, then of course we may think that modifications of these genes may generate morphological variation. Here I indicate may generate because I think still the clear-cut cases of clear demonstration of this is not very, very numerous cases, but I mean, it's a typical, it's sort of a typical statement where we all agree, but we still lack sort of body of evidence. It will come. Now I show you here this wonderful fly from which was produced by Ed Lewis. This is a fly that has 2 pairs of wings, which is quite unusual for Diptera. Diptera is only 2 wings. Okay. This fly has 2 pairs of wings because it has transformed the 3rd thoracic segment into the identity of the 2nd thoracic segment. And this picture I used to introduce 2 things. The fact that flies develop by making segments, slices, and then to identify these segments into different morphologies. Okay, and this is, I think, a very good, very nice illustration of this process. Now, the genes that instruct these different segments that you can see here, segments, to produce the proper morphology are these Hox genes that Claude mentioned in his introduction. And to make a long story short, this is this relation of polyneuropathy, which was actually worked out by Lewis, genetically speaking, on one of these clusters, the Vertebrates cluster, but he was right on almost everything. And you see here with this color code that once you have made these segments here, you will identify these segments by expressing these different genes, and you see these colors, they follow the order of the genes in the cluster. Okay? So a gene that is in red here, labial, will be working in this part of the body, and then in this one, this one, this one, this one. So there's sort of a correspondence between the order of the genes on the chromosome and the order of the structures that these genes determine in the adult. Okay? This is collinear relationship. Now, of course, vertebrates are also segmented, and I selected this very nice picture. This time it is a human fetus. And you see here these traces of segmentation. These are ribs. Okay. On the right-hand side, you see a little skeleton of a mouse. I removed the appendage skeletons, the limbs. And you see that we are built in a way which is very similar to flies. The process is a bit different. but we are also made out of a reiteration of segments, and these segments are different from one another. Okay. This segment here will give rise to a cervical vertebra. This segment here will give rise to a thoracic vertebra. The thoracic vertebra will bear a pair of ribs, whereas the cervical vertebra won't have any ribs. Okay. Now, an important difference is that this body plan here, unlike that of Drosophila, which is made by sort of a process of slicing something that exists, this body plan is made in a progressive way, starting here at the level of the neck and extending towards the posterior neck. Okay? So it's a process where you start making this part here, and then you add this one, and you add this one, and you add this one. And in vertebrates, it takes between 60 to 90 minutes, in fact, to produce the entire column. It follows a process that is referred to as the somatic clock, which was proposed by Jonathan Cook about 30 years ago, and the detail of this process have been quite well worked out by the lab of Olivier Pauquier when he was at the Star Wars Institute. And of course, much in the same way as in the flies, once you produce these segments, you have to identify them. You have to tell them, now, you make a cervical vertebrae, you make a thoracic vertebra. And much in the same way as in the fly, the genes that are responsible to do this are the Hox genes. There's a slight difference in mice, because you may know that vertebrates have been through 2 rounds of genome amplification, so we have 4 copies of these clusters. They are illustrated here, and you see here these color codes, which again, you have this, the order of the genes on the cluster, they illustrate the order of the structure in the mice. This is what I refer to as a molecular homunculus, in fact. It means that we have a sort of a preformed information in our chromosomes regarding our axial structures. Okay? It's not that these genes are scattered throughout the genome, that we have this collection of genes and they interact in trends, as many, many genes do, in networks and so on. In this particular case, there is a relationship in cis. It's a meta-cis relationship. Okay? And it's the fact that the genes are linked that gives instruction. So that tells you that we inherit from our parents a level of complexity that is already above the level of genes. This is organization of genes. It's a meta-cis system.
Well, you break the combination becomes unstable, right? Because if you break it, you just don't reinforce. Right?
I'm coming back to this in a minute. So, of course, I told you that vertebrates make their body in a temporal way. You start here, and it takes about 2 days to reach the end. So you need to activate these genes also following a temporal schedule, because if you activate the posterior genes at the time you make this part here, you will stop the body here. You will make the posterior here. Okay? So, if you look at the timing of activation, you see that the activation of the genes follow again the order of the chromosomes, and it's something that we refer to as the Hox clock, which every 60 to 90 minutes, there's an additional Hox genes that becomes activated during the development of the trunk. Now, for the past 15 years, as Claude mentioned, we've tried to understand how this system works. What is the mechanism that drives this sequential activation? Okay. Which mechanism can translate the DNA topology into time and space? Okay. You have to imagine that you have these 4 little images of your axial structures in the chromosome. They are very, very small. It's 100 kilobase. Yet they fix the timing of activation and the fate of the segments. Okay. How does it work? Now, we have approached this by a mixture of genetic and biochemical approach, and I'm certainly not going to go into the details, but I'd just like to give you the bottom line. We have used genetic tricks that allows you to recombine these genes. This is what we were mentioning. And in fact, if you would show this picture to a child and tell him, look, that comes first, second, third, fourth, Okay. What would you do to understand the system? I think many clever children would tell you, well, it's easy. You can remove number 3, for example. It's a little game of logic. If you remove number 3, now number 4 is at the virtual position of number 3. Okay? And the question you ask is the timing of number 4 or number 3? So in other words, is the timing of activation associated with the gene itself, or is it associated with the cluster structure? So is it a gene-specific feature, or is it a metasis-specific feature? Okay? Or another thing you can do, you can break the cluster into 2 pieces. That's what you were referring to. And then the question is, then the clock will start ticking here, and then there, and then what happens? Does it jump, or does it start again? Or, okay, this kind of question. And by doing this for about 15 years, actually, we ended up with a general framework, which is the following. We found out that the activation time of a given gene, of this clock, is function of its relative position within the cluster. It has nothing to do with the gene itself, to such a point that you can take any kind of promoter, Catabox, GC-Rich, any kind of gene which has nothing to do with development, and you trash it within this cluster, and immediately the gene will take over the timing of the cluster. It is completely gene independent. Okay? So if you are close to this part of the cluster, you will be activated rapidly. If you are far from this part, you will be activated late. And additional experiments suggested that, in fact, this regulation is fixed by 2 opposite types of influences. There is a positive influence coming from this part of the DNA. By positive influence, I mean things that try to activate. Okay? And there is a negative influence that comes from the other side. This is things that try to pack, to inhibit, to repress.
But biochemically, what they are?
I'm coming back to this. So there are 2 ways to see the system. If you look at this, you can say, well, they are essentially if you think of a logic, what you would yourself design if you would be in charge. Okay, there's 2 possible ways. Either you can activate this as many genes are activated in the genome, that is by a classical system of trends. Okay? For example, this gene would make a protein that would be necessary to activate this one, and the 2 proteins would activate the 3rd one, etc., etc. So a mechanism that is based on the activating limb, okay, on the green side, the mechanism to activate. Okay? Or you can think of a mechanism that is based on the red limb, that is a mechanism that represses everything and progressively releases. Okay? Now, the mechanism based on the green side is, to me, extremely difficult to realize, because you have 10 genes packed into 100 kilobase. You have to realize that 100 kilobase, the entire cluster would fit into an intron of a decent gene in vertebrates. You see the intron of the dystrophin, for example. We could base it. It's very, very tight. So it is, for me, extremely difficult to think of a system where you can activate these genes in trance without making mistakes. And mistakes are not acceptable in this system. If you make a mistake, you will change the morphology. You're dead. So you need a system that is much more tightly regulated. And of course, if you work on the red side of it, you can end up with a system that is much more tightly regulated. That is, imagine a system where you close the cluster and then you unfold it progressively such that you are sure that the genes are available one after the other in the correct order. Okay? So when the technology became available to look at these positive and negative structures, we had a look. And the first thing we did was to look at the presence of a mark, and without again going into the details, this is a Polycomb mark. That shows you where the negative molecule is, in fact. And what you see here is a piece of chromosome, and this strong blue here, it covers exactly the place of this cluster. Okay? You see that early on, before this point, before this phylotypic point, okay, in the early stages of embryogenesis, the cluster is covered with this negative mark, suggesting that nothing works. It's blocked. Okay, it's totally repressed.
It's methylated or what?
Sorry?
It's methylated or what it is?
It's a 3-methylation of lysine 27 on the histone H3 tail, in fact, which is achieved by one of the components of the Polycomb machinery. Okay. Now, if you look at the structure of this piece of DNA, the 3D structure, and this is done by using a methodology which is complex, and I don't go into the details, but if you look at the structure, you see that within this cluster, this is again the cluster, within this cluster, you see that every single part of the cluster contacts every other part of the cluster. For the expert in the audience, this is a chromosome conformation capture. It's a 4C sec to make it precise. And you see that if you take this viewpoint here, you contact everything. But as soon as you're out of the cluster, it's flat. Okay? If you take this other viewpoint here, all this viewpoint, all this viewpoint, all this viewpoint, regardless from where you look, okay, you are always contacting all the other points.
100 kilobytes.
It's 100 kilobytes.
Can do that.
So it means that the cluster is like this. Okay? Every single point contacts all the others. It's a sort of, it's a dense structure. Okay? Now what happens when you start activating? What happens when you approach this point and when you will start to activate these genes? And I show you one example here. That was the work of Natalia Soshnikova. You see that at the same time the transcripts are moving through the cluster, you have a retraction of these negative marks. Okay? So it means that along with the activation, you have a retraction, disappearance of this negative mark. Okay? What happens to the structure? It's exactly the same. If you look now midway through activation, so now the cluster is activated midway. Okay? Half of the genes are active, they work, and the other half is still inactive. Now you see that if you look from this viewpoint here, you have a domain here, but you no longer contact here, and here you no longer contact here. It means that whenever you are midway through the activation, you no longer have a cluster like this. You have two half clusters like this. Okay?
And a single passage or single RNA polymerase with that?
No. It cannot be because it goes backwards. Okay? You see what I mean? The gene orientation is opposite to the opening of the cluster. Why? Well, this is something I was totally not planning to say that, but we actually discovered this a couple of weeks ago. Why? Because that was a mystery for us. Why all these genes have the same direction of transcription, which is opposite to the opening? Well, for a very simple reason. If you have one that is like this, it opens the 3-methyl-K27, and you just, okay? But that was a parenthesis. So, the end result is what we've called the Rosary Mode. Okay? It is like a rosary. Do you all know what the rosary is?
Every morning.
No, I'm not a great expert in rosaries, admittedly. But I remember my grandma, she was constantly doing this thing, and she was a very Catholic woman. And rosary, yes.
Rosary.
Yeah, rosary. What do you think, a rosary?
I would say rosary.
No, a rosary. And she was a rosary is something where you have a different ave, and you shift one after the other from one hand to the other. Okay, and this is what happens with the genes. Okay. Now, of course, the problem is that we don't know what is the force. This is one thing we don't know. We have no idea what is the force. What is the thumb in this system? And we don't know what is the directionality. We have no idea why it starts there rather than there. We have some ideas on this, but so far we have no demonstration. Okay, but what we can see is that it works like a rosary. Okay, so early on you have this thing like this. It's totally packed. I had an Italian postdoc in the lab who called this a bunga bunga structure. Everyone contacts everyone. And then progressively you pull this thing out. Okay? And that has a lot of advantages. First, you are sure that the genes will come in the right order. You cannot make mistakes. You pull it out, so it's 1, 2, 3. You cannot get 7 before 6. Okay? Second, you can fix the system. You pull out 1, 2, 3, you are here, and then you have a system that freeze it. And then you go down and you pull out 4, you freeze. 5, you freeze. So it's a system that allows you to maintain, to memorize. Okay? And finally, it's a system that makes absolutely sure that the last one, the one which is at the very, very end, will come the last. And this is, of course, the requirement number 1, because the last one terminates. The last one is the stop. So if you activate the stop too early, you stop. Okay? So it's a system that works quite well. I found it interesting, if you look at this, I found it quite interesting to look at the semantic, because development, in fact, the root is Philou. I remember years ago I was invited in 20 years ago, I was invited in a meeting in Italy. It was a genetic society, Italian genetic society, and all the talks were in Italian. And for 3 days, I heard this guy saying, sviluppo, 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 sviluppo. And after the 3rd day, I said, please, what means development? He said, oh, thank you. But sviluppo is a very interesting word. Sviluppo, the root is the same root as envelope. Okay? It's something you open. It's a classical Aristotelian view of in fact, if you look at the German root, it's Entwicklung. Wickeln in German is to wind, to wind. Okay? So Entwicklung is to unwind. And Spanish is desarrollo, enrollar, it's desenrollar. These are all inspired by a classical Aristotelian view, in fact, that you have something preformed, and in a way you desenrollar. Okay, it's very interesting. Okay, but this system is extraordinarily difficult to evolve. If you think about it, it's a system where you can hardly think of a better way to implement this series of genes. You can think of transacting, protein floating around and so on. It's very, very difficult. And this is one of the reasons why I think it fixes this point of meta-constraint here. Okay? This is a very interesting point. In fact, as a sort of a metaphor, it's close to a metaphor. It's not quite a metaphor, but it's almost metaphoric. One could think that to go from there to there, you need to unwind this thing here, such that you can go through. You see the issue is halfway through a metaphor. Okay, you need to pull it through. That's 1, 2, 3. Okay. Now, this is a point where 2 very important mechanisms are meeting. It's 2 clocks. The 2 clocks are meeting. Okay. There's this Semitic clock, which I told you before. It's a clock, it's a segmentation clock. That's the clock that will make the segments. Okay? And then the Hawk's clock, the one I just described, is rosary stuff, which at the same time you make segments you will identify the segments. Okay. Now, of course, these two clocks, they must be very, very well coordinated, at least within the same species. But they can also be a very rich source of variation, of course, because if you introduce offset heterochronies into these two clocks, you will immediately change the morphologies. And a very good example is the comparison between chick and mice with the cervical vertebrae. Okay? You can perfectly imagine here that rather than having 7 vertebrae, if this clock ticks 7 times, if the segmentation clock ticks 7 times, without having the Hox clock ticking, you simply add 7 segments here. Okay? So you have to see the system like, you know, you get these embryos doing a little bit what they want, making epibole, or making cleavages and so on, and suddenly they converge towards this point because they have to go through these two mechanisms, this meeting of the two clocks, and then they go tack, 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 tack for 2 days. Hop, and then they can diverge again.
But this is for vertebrates, yeah, for invertebrates it's different.
This is for vertebrates.
Yeah, vertebrates.
There are some issues, you know, whether that exists for. There's been quite a lot of paper ever since, and it seems that it's also the case in other taxons, but not in all. Okay. Now this big jump here, of course, is in a way not easy to reconcile with the strict Darwinist view of gradualism in fact, because you can introduce very substantial changes by playing around minimal force, okay? And that, of course, raises the question of macroevolution and the idea of constraints, okay? Does it exist? Or now, I'd like to spend the last 50 minutes telling you about this, and because it's something I'm interested in, of course, let me start by reminding you of a little bit of the theory, or unless you are all very familiar to it. But let me just tell you 2 things. First, the theory of evolution is not Darwin's. Darwin is one component of the theory of evolution. The theory of evolution emerged much before. Notably, Darwin was very much influenced both by Lamarck, of course. I remember that on the beagle, more than 50% of the species he identified in the beagle, if you read his notebook, were identified because of the book of Lamarck, okay, the Traité des Insectes, which was the only book available at that time for the invertebrates. Okay, Lamarck was the king of invertebrates. Okay, and of course by Malthus. Now, unfortunately, Lamarck had not read Malthus. Okay, it was too late. Darwin had time to read Malthus, and I think this combination of influence led to the formulation of the theory. But theory is based on 2 limbs. Okay, first, transformism. That's clear. Animals derive from animals. Okay, and this has nothing to do with Darwin. This is purely Lamarckian theory. That's his major speech in the museum in Paris in 1800. Okay, where he brings the basis of transformism. Okay, animal evolved due to particular processes. This is the contribution of Darwin and Wallace. Actually, for those of you interested, if you read the letter that Darwin and Wallace sent in 1858 to the Linnean Society announcing the book. Okay, I mean, you know the story. Wallace writes to Darwin and says, oh gosh, I'm going to be scooped. So let's write a letter to the Linnean Society telling them, hey, wait a minute, next year I'll send you a book on this. The first reference that Darwin makes is to Auguste Pyramus de Candolle. Auguste Pyramus de Candolle was a scientist in Geneva, another emigrant, French emigrant, who was the first to state that nature is at war. And the guy has realized this by looking at his garden. He had realized that when you grow plants, there are always plants that invade more than others. And he actually visited Darwin in 1935, I think, much before Darwin had realized the theory. So what I want to say is that Darwin brought a fantastic piece of the theory, but it's not at the origin of the theory. It's only one major contribution to it. Okay? Now, variation, how? Organisms are subject to variation. Selection, why? The best adapted variation will be maintained and propagated. That's the how question and the why question. Now, if you look at evolutionists for the past 60 years, or ever since neo-Darwinism has been formulated, the how question has been completely neglected. Of course, when you don't know how to answer the question, you neglect the question. But people have mostly cared about the why question. Now, the how question has been largely ignored or hidden by what I think the sort of lethal weapon of evolutionary time. Okay? If you talk to classical evolutionists, I hope there are some in the audience, but they will tell you, well, if you have enough time, you can essentially do everything. Okay? And that totally ignores the notion of constraints. And the other view would be to say, well, even if I give you billions of years, there are things that you will not be able to produce as a natural variation. Okay? These are the internalist side of the theory. Okay? Now, internalist theories, they refer to the existence of internal constraints. Okay? So there we are. Now, these internal constraints, they can be within an evolving system, but they can be rational or they can be irrational. Okay? And in this respect, intelligent design, for example, can be somehow considered as an internalist theory. And this is what it is so difficult for us to have real scientific discussions on this, because it's mixed between these things. Okay? To talk about constraint, immediately there are people who will say, hey, you are against Darwin and you give support to intelligent design and so on, which is, of course, a very constrained theory.
What is the criteria of rational exactly?
I would say a rational internalism is something that we should be able to demonstrate, or at least that should be able to demonstrate that it doesn't exist. Unlike what I would call an irrational internalism, like Bergson, for example, the élan créatif, or things like this. Okay, but let me continue. So these constraints, they would restrict the realm of potential variation. This is, that would give an apparent teleonomy to the system. Of course, it's not a teleonomy; it's an apparent teleonomy. It means that the number of solutions you have for variation are restricted to some extent. It can be lightly restricted, or it can be enormously restricted. Okay. Now, this is a quotation I got from Pere Alberge. Pere Alberge was a collaborator of Stephen Jay Gould in the museum in Harvard, and he passed away, unfortunately, after moving back to Madrid. He worked a lot on this, and he said, I refer to this philosophical tradition as internalist. Okay? But look, the first references he gave was Bergson 1907. Bergson is Evolution Créatrice. This is what I call, it's quite close to intelligent design. In fact. Not exactly, but quite close, in fact. Pere Alberge didn't believe in genes, in fact. I had very interesting discussions with him. He didn't believe that genes would be important for development or for evolution. Okay? Now, this confusion between rational and philosophical internalism has made this way of thinking somewhat suspicious. That's what I was just saying. Even though it was documented by Bateson and Lancaster and so on, the British School of Merits, which you may have heard about in the late 19th century, and Ray Lancaster actually proposed a whole set of rules. This is rules of metamerism, okay, in vertebrates. These are things you can do, things you can't do. He made a classification of these rules. These are complicated wording, so don't read them. But for example, you will never find a single animal that will have a vertebra in the middle of the thorax that has no ribs. It's a rule, okay? And you can go to the museum in Paris, in the paleontology gallery of paleontology, you can screen thousands of animals. You will never find an animal that has rib, 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 no rib, 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 rib. Okay, it's a rule, and you have a set of rules like this. Okay, so there are constraints, and Bateson, who invented the terms homeosis, of course, had listed some of these constraints. In fact, interestingly enough, if you look at development, not evolution, development, constraints are very well accepted. We are probably the only solution to the problem of the embryo. That's what Volpert claims in his book. Okay? So we are the result of a maximal constraint. That is the only solution, it's us. Okay? Waddington, in 1942, with this picture of this epigenetic landscape, which we see now in every single seminars when people talk about chromatin modification and so on, to, you know, up to here. But the interesting thing in this picture, the interesting thing is that it shows you that during development is nothing but constraints. In fact, you start here and then you take a decision. And okay, now the fact to understand the nature of this constraint in development is interesting. But you hear Stefan Behling view of this, you know, reprogramming and deprogramming and jumping from one to the other. These are simply giving identities to constraints, in fact. But during development, they are well accepted. Okay? During evolution, it's very different. I show you here two pictures. It's again the picture of a fly with 4 wings at Lewis. And here a picture which was produced by the lab of Walter Gehring, with these additional eyes, and I'm sure you must have seen this in the press and so on. These additional eyes are produced by the activation of a single gene. Okay, it's not so important to know which one, but single gene. And of course, when you look at this, when you look at that, you can see, wow, it's amazing that a single gene can do a full eye.
The wrong segment. It's gene in the wrong segment, yeah.
It's a gene that is controlling eye development. Ask your neighbor, he knows better than me what it is. But the point is that activating a single gene at the wrong position can give you an additional eye, okay? Now, you can turn around the question and say, well, it's not so amazing, in fact. Well, what is amazing is that it always does an eye. Okay? Well, it never does anything else. It's not the fact that it can do it. It's the fact that it cannot do anything else. Okay? It's maximal constraint. You start something and there you are. You don't think about, should I go this? No. You just go into it. And that illustrates that we are made out of an addition of constraints.
And then the gene is silent. After development, the gene becomes silent.
Becomes?
Silent. It's not functional. Silent.
I don't know how long the gene must work to get the eye properly done, but I think a very short time. What counts is what comes after. Okay, so you can, in a way, represent the participation of externalism versus internalism in nowadays view of the theory. In fact, if you would consider it as, let's take an average in the scientist population, you would say that most of the strength of evolution is based on externalism. Okay, things that come from the outside, classical natural selection. Okay, gradualism. But gradualism, I wrote here, not a la Darwin, because Darwin was much more careful, in fact, than people like Ernst Mayr or Dobianski, who were really orthodox in the way they would see gradualism. Darwin was much more careful in his writing. And a little bit of internalism here, okay? I wrote a la Goldschmidt, you know, the hopeful monsters of Goldschmidt, or Bateson, people who think that at some point during evolution you can make big jumps. Okay? Now you can say, well, how come? Because making a big jump, it's totally helpless. If only one make a big jump, you need at least to have two making a big jump if you want to. Okay? But you can start thinking that sometimes you reach the genetic equilibrium that is close to one solution. So it will repeatedly move into one solution.
Because some biggest jump very common, doubling into genome is very common.
Yeah.
It's
For
very
example.
common.
Okay.
The biggest jump you can make.
So perhaps we could reevaluate this, and in the light of what we have recently discovered over the past 30 years. And let me go very rapidly to what I think are the three major discoveries in this field, okay, for the past many years. This is what Claude mentioned, actually. First, the fact that genes are universals, and I'm always amazed to see how rapidly it went into our textbook, and now we all consider that this is, you know, a common knowledge, and the universality of principles, and the universality of genome, because after all, if you compare the trout chromosomes with our chromosomes, you find synteny of 30 megabase. Okay, it's insane. So we have the same chromosome as trouts, you know, in fact. Okay. There are a few differences, but not much. Now, the question is, to what extent that changed the paradigm, okay? Well, I remember when I studied, to the question, how do elephants get the trump? We would say, well, because they have trump genes. And of course, for you guys, you think it's insane. But this was 40 years ago, okay? And now we phrase the question quite differently. We say, how come we don't have a trump? We still have the same genes as elephants. Okay? It's a very big change in the paradigm, in fact. Now, then came human sequencing, and that was a big shock. As you know, nematodes, 950 cells, 15,000 genes. Human beings, something like 10 to the 14s, 10 to the 15s, no one really knows, 20,000 genes. I do remember, and I'm sure some of you also remember, that before human sequencing, we would estimate the number of genes to 130, 140,000, you remember? And I do remember when this first figure came out, journalists calling me in Geneva and saying, but you must be depressed. You must be depressed. How can that be? Okay. And they were quite right, actually. Not that I was depressed, but how to explain that? How to explain that? Okay, that we have barely more genes than this little miserable animal. No, 300, 350. No, it's a great animal. But it's 350 neurons. No, isn't it? I mean, it's, you know. Anyway, because of pleiotropy, of course, that's the solution. Multifunctionality, pleiotropy. We use our genes much more efficient way. The way we combine them, the way we use them, etc. So we ask for much more per gene than the nematode. Okay? And you can see that now with the structures of the regulatory circuitry. If you compare between vertebrates and nematodes or flies, it's totally obvious. Okay, now we understand. But that has a cost. Because if you conserve the genes and genomes, okay, that implies that you need to do more with the same, as I said. You introduce multifunctionality, complexification of protein, regulation, networks, resistance to variation. Okay? So the more job you give to every single gene, the more difficult it becomes to change. Okay? That's the logical way of thinking. Okay? So you progressively constrain the system, and it leads to a higher stability and heavier constraints in what we can qualify as complex system. But I know it's difficult to really give a definition of what is a complex animal versus, you know, I wouldn't say it's C. elegans is. How many people work with C. elegans? That it's not a complex. It's not a complex animal. So perhaps at some point we have to revise this balance between externalism and internalism, and have something more looking like this, which is that there's a lot of externalism in very simple organisms like bacteria. evolutionists always come to us and say, well, look, we can demonstrate evolution because if you pipette antibiotics in bacteria, then the next morning you have a new population of bacteria. You have a response to something coming from the environment. That's externalism 100%. But these are bacteria. Okay? If you do it with rabbits, it's not going to work. Okay? So perhaps the internalism has more weight in complex animals, and perhaps evolution goes from natural selection to constrained variation, along with the complexification of organisms. That means that the respective importance of these two phenomena would change depending on the nature of the animals. Okay, from a complete externalism of bacteria to a complete internalism to complex system. Okay, so we would perhaps see a progressive transfer of the driving force from the selection towards the variation. And this is, in fact, if you think about it, it's a very politically incorrect statement. Amongst evolutionists, I'd like to be a little bit provocative with this. And the sense that variation is no longer infinite, okay, even over evolutionary times, but dictated by the nature of the system. Of course, we could likely add a trump to human, but in this case, we would be like elephants. Okay. No, it's an important statement to understand. We could probably add a trump, but then we would become elephant, because you cannot produce a trump without having the other stuff that comes along.
Well, this is a communal system because half of the brain of the elephant occupied with the trunk, yes?
Right.
You have sort of many muscles you have to control.
Right. So, the flexibility to develop highly adaptive traits is in itself constrained by the set of fundamental rules of the system. Okay? So once you have passed through this phylogenetic bottleneck, then of course, once you have passed through this maximal constraint, you are allowed to diverge a little bit, but not so much, in fact. You build on these constraints that exist. And this is what I would call the post-phylogenetic bottleneck. It's interesting from an epistemological viewpoint, because of course, it impacts a lot to me on the concept of model system. And when I visit the institute, like I had the chance to do today, I always talk to people who tell me, I don't think it was the case today actually, but who tell me, well, I'm working with this animal because it has the following advantages. And I remember in the early days of the fish, you would see people coming with a list of advantages, you know, as if animals would have advantages, okay? For example, the fly developed rapidly, or you know, C. elegans has very few cells. We know all the lineage. Great, fine, but can that be qualified as an advantage over what? The problem is that what we want to understand are the fundamental rules, the core of the rules. Okay, and each time you produce an advantage, it's usually an adaptive trait, very highly adaptive trait. It's something that diverges from the core of the central rules. It's, for example, the high speed of development in Drosophila. It's, for example, the cell lineage in C. elegans. It's regeneration in neurodeals and so on. Okay? So usually we select our model systems because they allow us to study something which diverges from the core, from the set of basic rules, okay? And we systematically hope that we will have a centripede reward, okay? That by understanding this, we will come back to the central rules. But in fact, what happens is quite the opposite. In fact, we work on centrifuge things. This is the more we understand an adaptive trait, the more we go into the adaptive trait. So, we may as well select the model system that is the less appropriate for any problem we would like to solve. In fact, epistemologically speaking, that would probably be the best solution. Because as soon as you select a model system, you hit the constraint. Okay? Now let me terminate by telling you why I think it is so difficult for evolutionary biologists and for us to some extent to discuss about these things, to discuss about constraints, about internalism and so on. The first thing is that an orthodox gradualist view, like that of Dostoevsky or Ernst Mayr, explains perfection. Okay, explain how perfection can be reached, and this is something we are very, very attracted to. Okay. We would like to see ourselves as the summum of perfection, and if you come and say, look, biology forbids perfection, it's a very difficult thing to admit. Of course, we all think we can admit it and so on, but we are not alone. Okay, we're living with hundreds of centuries on our back, and this is something we have problems to admit. Okay. Internal constraints make perfection impossible. It makes the selection on a genetic equilibrium rather than on a collection of perfect traits. Okay, so we would need to admit that we are very average solutions. Okay, we are in fact a collection of imperfect things, but the wall works quite well actually. The sort of compromise. The compromise works quite well. Okay, so the less solution to be selected for, the less importance given to notions like fitness, adaptive value, which are very often of anthropomorphic nature. Okay, and I indicate this. We love Malthus. We love Lamarck. Okay, if you look at what, I mean, if you go to meetings on epigenetics these days, you see it's Lamarck is coming back. It's Lamarck everywhere because it is so pleasing for us to think that we could influence our genes. Okay, that if we behave well, we get some reward. You see, if we do things properly, our children will benefit from it. It's all the social insurance system, in fact, transposed to genetics. Okay? Those who behave well, they will transmit something to. We are bound to like Lamarck, in fact. And finally, internal constraint during development. Of course, there is questions on free will, and this is something that's extremely difficult to admit. If we admit that we are built on internal constraints, then our brain is built on internal constraint, then the conclusion is that free will is a myth. We are absolutely not free to think what we think. And in fact, this is the extreme position of Spinoza, as you may know, that mental processes are of corporeal nature. Okay? It's the famous substans cogitans, substantia extensa, so the content and the container. Okay? It's one and the same thing, depending on how you consider it. But it means that you can only think with the structure you have evolved to think. Okay? And to think that you are free to think is just a myth. Okay? Now, interestingly enough, that's my last slide. Darwin had thought about it, in fact. And if you look at, I think it's chapter 13, he says, the general delusion about free will obvious, because man has power of action and he can seldom analyze his materials. Originally mostly instinctive, he thinks they have none. Okay? And he terminates by saying his views should teach us profound humility. One deserves no credit for anything. I think from his part, it is really a fantastic statement, nor how one to blame others. Thank you very much.
If you look at genes, from, let's say, from a nematode or a human being, can you know which genes are human and which are human?
You mean if you would be an alien coming into the Earth? And, yeah. I think if you would take all the genes of C. elegans and all the genes of human, and you would be an intelligent alien and make an algorithm and so on, you could. But if you only look at the coding regions, at the protein regions, in a sort of a first glance, you would not.
There are no particular thing in C. elegans that would say, oh, this is a C. elegans genes. No.
Okay, and then I have one more quick question. When you say the eye, you know, made the eye, I mean the gene, like, how does the gene know it's done its job and made the eye? Or can you activate it to make another eye if something happens to the
But the gene doesn't know nothing. Genes know nothing.
No, but you know what I'm saying.
Yeah.
In other words, how does the gene know it's done its task? The
Yeah.
mechanism.
I don't think there are mechanisms like this. There are mechanisms of control. There are mechanisms that...
That's what I'm asking.
Right. Mechanisms that come after and control that the stuff has been correctly done. Yes.
Is there a potential for to reactivate that gene to make another eye should it be necessary?
You mean, for example, for medical purposes,
yes.
to make it?
Yes.
Well, yes. If you can make an eye during development, it's not excluded that you can, let's say, regenerate part of it or produce a piece of cornea or something like this. Now, to make a brand new eye, okay, that would be very difficult because the eye is induced by very early mechanisms in the brain and so on. So no, but to regenerate part of it, I'm sure it's going to be feasible sooner or later.
Sasa just remade an eye in vitro. It's an illness, the retina in vitro. So, I mean, it's getting closer.
Yeah.
Questions? So I have a question for you. So you mentioned that as you become more sophisticated organisms with more cells, the same number of genes, we get our constraint to be much, much tighter. However, if you look at the vertebrates, for example, the diversity is quite extensive. I mean, an elephant or a mouse don't look exactly the same, which means they still have a large set of parameters they can vary to be able to get this huge amount of diversity.
Yep. Well, you say they are very different. They aren't. Are they? No, really.
But you think there is more? I mean, you expect to have more diversity among simpler organisms which have fewer cells, fewer constraints, and the same number of genes. Is it a correct statement or not?
No, I think it's correct. But this is my bias. I don't see an elephant being very different from a mouse because I see them at these transition points, and they are virtually undistinguishable from one another. After, what comes after is, it's details. It's having,
Because there's nothing that looks more like a ceiling and another ceiling.
No, I think you should really, once you have the chance, you should go to the Grande Galerie, the Paléontologie in Paris, and you will understand what I mean by this. If you look at the skeletons, if you spend 2 hours in this room, after 2 hours you confuse everything. They're all the same. They're all the same animal.
So I work on invertebrates, and so I agree with what you're saying for vertebrates, and it's sort of it's really a subphyla at the big stage, right? Because it's a chordate, they're chordate phyla, and it's hard to find that same stage in some of the other chordates. And so, do you really think that there's a phyla at the big stage? I think it works very well for vertebrates, but I don't think it works very well when you go to echinoderms or hemichordates or some of the other.
Well, the problem with them, there are several problems. I mean, echinoderms, there are very few. So to identify these stages, you need to have a bunch of species, of course. And whenever it's been tried, not based on morphological, because now it's no longer made on morphological characters, but it's made on sequencing RNAs. That is the work of different groups have done that. And it seems that it is correct in Diptera as well. The problem is that you need a minimal amount of species that are well characterized or characterized well enough that you can identify this stage. Okay? If you go to sea urchins or
we have sea urchin, sea star, sea cucumber. There's actually quite a bit of work on lilies. It's just that.
But all these species they've been classified into the same taxon because of some kind of morphological identities, which are not in the adult stage, generally.
Yeah.
And not in the very early stage, because that was not even visible at the time they made the classification. So it's something in between.
larval stage or something like this.
So I have kind of a two-part question. Also, very great talk. I enjoyed this tremendously for the time. So do you feel that with an intrinsic view of evolution, it gives you a more predictable model for development and evolution of species in general? And could this be predictive of the number of species of each type that you see?
Absolutely.
Which is, I mean, we can fairly measure that. You know.
So people, I mean, I'm not a modelist or a mathematician and so on, but I have colleagues who argue that once the embryo will be computed, then we will know what will be the next step. Okay, because it will turn obvious that a few solutions will be available, not a lot, a few solutions. Pere Alberch, actually that's really interesting stuff. Pere Alberch, he wrote. He was an expert. That's why he came back to Madrid. He was an expert in mirror painting, and he wrote a very interesting book on the fact that Miro had spent all his life to paint the same symbol. He could never escape from the same symbol, little symbols. Okay, this is always a constraint. And Pere Alberch had made a search in when he was in Harvard at gynecology. He looked at human monsters. Okay, that was the start of his thinking on constraint. He looked at this collection of human monsters you have in medical faculties, and he had noticed very important thing. First, nature never make something that you cannot identify. Very interesting. It's always something you can identify. If there's an extra piece, it's always an extra something. It's an extra leg, it's an extra limb, it's an extra head. It's never an extra nothing. You see what I mean? And second, he had noticed something very interesting. His paper is called The Logic of Monsters, actually. He noticed that depending on where the extra thing appeared, it changes the identification. For example, if you have an extra limb here, it's always an arm, never a leg. If you have an extra limb here, it's always a leg, never an arm. You may think it's logical. No, it's not logical. It means that when you start something, it's constrained. You know exactly what you're going to do, much in the same way as Pax will induce an eye. Okay? And it's from this teratology analysis that he developed this idea of constraint.
Yeah.
I think the philosophical stuff at the end was amazing, but I want to talk a tiny bit about the mechanism of timing that you referred to for the Hox clusters. And you said that if you can delay timing, then you can have more cervical vertebrae or something like that.
Right.
What is the mechanism of the timing? Is it more DNA between the genes? Is it, what do you know?
No. So there are things we only know what is not, okay, but we don't know the answer. So we know it's not the absolute distance between genes. For example, you can duplicate a non-genic region, doesn't change. We know what is key is the relative position of genes. Okay. I remember in 1993 already, when we started to make gene transpositions, Ben Lewin, for those of you, Ben Lewin had told me you should introduce junk DNA into clusters. And I replied, but what is junk DNA? That was the end of our conversation. But it's very, very delicate. What would you introduce to see it there? I
just wondered if
No,
the
but
difference between
the dream,
species. Yeah,
the
exactly.
dream experiment would be, to introduce something like 50 kb, a good 50 kb of, let's say, sort of inert DNA you would find, somewhere not too far nearby. Okay, and you would introduce this big piece, but it's very difficult to do.
So I have a question which is related to that. So it seems that the hot gene takes advantage of this linear collinearity. And why is this only set of genes in biology that basically takes advantage of the one dimension of the DNA?
No, it's not the only one. No, there is a cluster of...
there is a globin genes?
No, the globin works differently. No, there is another cluster in the sperm, which is...
But yeah, the raw, okay. But it's not something which is widespread. You have one dimension that nature doesn't exploit.
But why would that be?
It's a great system. I don't know.
But to do what? To do what?
I don't know. You expand to limit the constraints.
But to do what? You have to specify this axis, you do it with the hogs. This axis, you do it with the hogs. The head, you do it with the hogs. The gut, you do it with the hogs. Oh, what is that?
But you say you will always do it with the hogs because it's the best way to do it. It's one solution. Why can nature evolve another solution to something?
Ask nature.
Yeah, I think it was very handy. And at the time limb emerged, then it...
She's the answer.
Well, we also continue to develop in time as we go through stages of development. Maybe genes controlling transitions to larval stages, we might find have this linear organization.
Thank you.
Just that idea.
Thank you.
Could be.
What's the question? Are you overwhelmed by?
There was a question way back in the left.
There was a question here also.
Yes, she said her arm up for like
I was
15
just
minutes.
wondering if you know which genes are controlled by hogs. Genes? Or
Which genes are controlled downstream of the hogs? Oh, this, yeah, you know, there's been many work done on this, like differential screen and you know, gain of function and gene chip and sequencing and so on. And we isolated a few, and other people isolated a few, but nothing that, you know, it's so crazy and important that I would be able to give you a brilliant answer. I think these genes, they work very differently. They work by adjusting balances, okay? You know, if you look in the limbs, if you make a mutation, rather than having this, you will have that, or then that, or that. So it's not genes which will trigger the activation of brand new targets or whatever. They adjust quantitative balances, okay? And it's very difficult to deal with these kind of problems, you know? When you change 10% of 60 genes, it's much more difficult than when you change 100% for 2 genes. So I would say this is still something that, you know, it's not our direct interest, I must say. I think this, I mean, don't take it wrong, but this target stuff, for me, is rather pedestrian in the sense, I don't think it's going to be very different from other genetic systems that exist, okay? There'll be transcription factors and target genes. And we are more interested by the regulation aspect.
Yeah.
Going back to the idea of constraints and one of the internalistic ideas where you would have major jumps between species at a time. Have we ever been able to try and break through some of these barriers by like trying to mimic Hogs gene implications or something like that?
You mean to try and reproduce evolutionary transition by having a genetic approach? No, I'm not aware. There's been a lot of tentative. You know, I was telling to someone, I'm not sure this person is there, but something interesting, which is, generally speaking, in animals, you can make any kind of genetics. You always remove things or reduce things. It's very, very difficult to add things or to enlarge things. It's very strange. As if we would reach a sort of entelechia, you know, Aristotelian entelechia, sort of the level of, you know, that we have achieved a sort of level of plenitude that you can remove from it, but you can never go more.
So we have reached perfection.
No, it's the entelechie. It's not a perfection. It's a sort of a plenitude. It's the place where we should be.